So I think we're ready to start this parallel session. So I'll hand over to the two chairs to kick off so we can get a prompt start. Thank you.
Thank you.
Yes, okay. Can you hear me now? Yes?
Yeah.
So my name is Sandrine Humbert. I am a basic scientist interested by Huntington function and the dysfunction during the pathology. So as we have heard several times, Huntington disease is for most cases late manifesting. However, the disease driving protein, mutant Huntington, is expressed since the first days of life during embryogenesis. And this started to raise the question of whether there may be a neurodevelopmental component in HD. So this neurodevelopmental component is supported by studies in mouse that are showing abnormalities in brain development in HD, but also by early pathological signs in patients for which the most probable explanation is abnormal brain development. So I'm really very happy to chair this session that will address some aspects of this topic. Cortical and striatal development using fetal samples, mouse models and cells, and pediatric HD that may represent neurodevelopmental delay. And my co-chair is Sylvia Gines, and she will introduce herself and introduce the first speaker. Thank you, Sylvia.
Okay, thank you so much, Sandrine. It's a pleasure to be here. Even it's online meeting, so hopefully next year we'll see each other in Bologna. So I'm working since many years in Huntington disease in the University of Barcelona. So now, because we have really short time, just move rapidly to the first talk. So the first talk is given by Dr. Monia Barnat. She's has been working. She's a senior investigator in the Grenoble Institute of Neuroscience, and she has been working in neurodevelopment and in HD for many years. I think that since her PhD and postdoc, she's working on neurodevelopment in HD. And now, as a senior investigator, he's going to explain today how mutant Huntington is modifying the development of cortex and how these alterations can really go to the pathogenesis in Huntington disease. So, Monia, we cannot hear you. Okay.
It's okay now?
Yeah.
Thank you.
Thank you.
Thank you. So I will share my. So thank you for this introduction. So it's a great pleasure for me to present this data. So I am going to talk to you today about how brain development, in particular the development of the cortex cerebral, is the cerebral cortex is affected in Huntington disease. It doesn't work. So, as we saw during this meeting, Huntington disease is a neurological disorder caused by the mutation in the gene encoding for Huntington protein and leading to degeneration of brain regions, in particular the cortex and the striatum. So it is important to know that this mutation will lead not only to a gain of a new toxic function of the mutant protein, but also to the modification of its normal functions. So what are these normal functions? So at a molecular level, Huntington protein acts as a scaffold and allow the formation of protein complexes involved in different cellular processes, including cell division and vesicular transport. So one well-studied example is the vesicular transport of the neurotrophic factor BDNF. So in normal conditions, Huntington regulates the transport of the BDNF along the cortical neurons. So from the cortex to the striatum, and this mechanisms is impaired in disease conditions, leading to massive neurodegenerations. So although Huntington disease is a late manifesting disorder in most cases, growing evidences, including neuroimaging studies on presymptomatic mutation carriers, presenting a decrease of brain volume, suggest that Huntington disease might affect neurodevelopment. And recently, this idea was corroborated in mice by the fact that the mutation restricted to the developmental window is sufficient to induce phenotype characteristic of HD in adults. So in the lab, we focused on the cerebral cortex, structure affected by a massive neurodegeneration. So as we can see here. So the cortex regulates the sensory, the cognitive, and motor function, and is formed of 6 layers containing mainly excitatory neurons. And the nature of these neurons depends first on the layer, but also to the brain regions to which they project. And via this neuronal connection, the cortex regulates these functions which are impaired in disease. So how does the cortex develop? So the cortex development starts early during embryonic life in a human and mouse, and consists on highly coordinated events resulting in the laminar organization in adults. So the cortical layer is produced by successive waves of neuronal migrations in an inside-out pattern, which means that the deepest layer is formed first and the uppermost layer next. So during the formation of each layer, we find the same cellular steps. So first, in the ventricular zone here, bordering the ventricle, we find the cortical progenitor, so the precursor of excitatory neurons. They divide by successive division, mitosis, and the number of these mitoses is regulated by a particular movement in the VISI called the interkinetic nuclear migrations. So it is a periodic movement back and forth in the VISI and allows to restrict and regulate the division at the ventricular surface. So during the formation of each layer, once the number of progenitor is reached, they differentiate into neurons. These newborn neurons migrate along the neocortex to reach their final destinations in the future cortex, where they undergo morphological and functional maturations. So in the lab, we have different mice models to mimic the disease, either expressing mutant Huntington or no longer expressing wild-type Huntington. So on these mice, we use a technique of the in utero electrophorations, which allow expressing protein of interest, in particular fluorescent protein, such as GFP, in embryonic cortex to follow the cell fate of progenitor and neurons in control and disease uterine environment development. Using this approach, we have shown the role of Huntington in different stages of cortical development and how these steps are affected in HD. So first, during embryonic life, we have shown that Huntington mutation and deletions affect the division of progenitor cells, leading to a decrease of the number, the generated number. And then the migration of these newborn neurons is also affected by the mutation and the deletion of Huntington, as shown here in the developing cortex, leading to cortical lamination and dendritic morphology defects in young adults. In sum, we have shown in HD mouse model defects in cortical development at different stages. And the next question was to know if this abnormality exists during cortical development in humans. So to address these questions, we analyzed healthy and HD mutation carriers fetuses in collaboration with Alexandra Dur's team in Paris, with all relevant ethical regulation and approved by the French Biomedicine Agency. So HD fetuses are obtained from medical termination of pregnancy after a prenatal diagnosis, and the control fetuses are obtained after a voluntary one. So we analyzed fetuses at gestational week 13, and at this stage, we had access to the early steps of cortical development, and we thus focused on the cortical progenitor in the vision. So here, as first steps, we analyzed the expression and the localization of Huntington and mutant Huntington in both conditions. So in control fetuses, we found that Huntington is expressed in the whole cortex, but in HD condition, the mutant Huntington was strongly accumulated at the ventricular surface. What does this accumulation area correspond to? It is the apical end feet of cortical progenitor, and these apical end feet are very important structure during development, cortical development, because they ensure the integrity of ventricular surface via junctional complexes, including the protein PAR3 and ZO1. So after analysis and compared to the control situation condition, we found defects in this protein, and this defect is characterized by a loss of PAR3 expression at the ventricular surface associated with a strong accumulation of ZO1, showing that Huntington mutation leads to defects in physiology of cortical progenitors. An important feature of this apical end feet is the primary cilium, as shown here, which sends signal from the ventricle and regulate the fate of cortical progenitors, which means proliferation versus differentiation. We observe that in HD model, defects in ciliogenesis, and these defects are characterized by a significant increase of cilia length and cilia density, as shown here in these images in red. And one of the mechanisms highly dependent on ciliogenesis and cell adhesion during cortical development is interkinetic nuclear migration, which is, as I said, a periodic movement in the VZ back and forth to regulate the number of progenitors. So to study this movement, we returned to a mouse model, and we electroporated in utero mouse embryo to express protein of interest, here the GFP. And after 2 days of uterine development, we analyze their dynamic behavior, so for GFP positive cells, on brain slices kept alive in culture, as shown in this movie. We found that in HD embryo, that the migration, velocities migration is significantly decreased in both directions, and that this slowdown is associated with a decrease in dividing cells, staying here in red in control and disease conditions. Another important parameter of the cilium during cortex development is its orientation. An apical cilia directed toward the ventricle, or a basal cilia directed toward the cortex. And it is known that an apical cilium is associated with a proliferative state of cortical progenitor, whereas the basal cilia is associated with differentiated one. So after the analysis of this orientation on brain slices in control and HD brain slices, we find a higher proportion of basal cilia in HD fetuses, showing that progenitor engage more rapidly in the neuronal lineage at the expense of their proliferations. So to complete these results and to have a readout, we stain proliferating progenitor expressing the PAX6 protein here in purple, and the differentiated ones expressing TBR2 proteins in green. And we find, after analysis, more progenitors engage in the neuronal lineage in HD fetuses. These results, if added to the decrease of cell division described before, suggest in fine a decrease in the number of generated neurons in disease conditions. In conclusion, our data reinforce the idea of a developmental contribution to HT, which will require a better understanding of the developmental mechanisms. And as in the mouse model, which is a robust model to study the molecular and cellular mechanisms, our results show abnormality in human cortical development. However, what is the significance of this? So further study are needed to know if these developmental defects contribute to the onset of prodromal sag and the vulnerability of neurons during the symptomatic phase, and how compensatory mechanisms prevent the onset of symptoms over several years. So thank you very much for your attention. And I would like to thank all the members of the Sandrine Imbert's team, all the patients and their family, and all our collaborators, without whom the study would not have been possible. Thank you.
Okay, thank you so much, Monia, for this talk. So now we have, I think, 5 minutes for questions. So I'm going to start with this question. So just waiting for questions coming in. So I think that you raised at the end really important questions, meaning that how these alterations during the development can impact in the adulthood. So what is known about the structure of the cortex in the adulthood in Huntington's disease? So how these alterations during development really impact at the end?
So it's not yet known what impact these developmental differences have on brain functioning on adults, but there are several studies showing that, for example, in HD mouse model, an alteration of dendritic morphology and also electrophysiological properties. So I think that the functioning of the neuronal function is disturbed during the first part of life, but there is some compensatory mechanisms that could compensate these defects, but after a certain time, we have a deregulation of these compensations.
Okay, so I think that we don't have time for more questions, so we need to move forward. So Sandrine, please.